Welcome to our WebCheminar on Mechanochemistry today. I would like to thank you for joining us today. My name is Stephanie Baumann, and I'm Executive Editor for the Thieme Chemistry journals, Synthesis, SYNLETT, Organic Materials, and SynOpen. Yeah, today we have a web seminar on mechanochemistry. So why? We have initiated a cluster, a SYNLETT cluster last year with Tom Robis as guest editor. And he has invited Chef Moore to join us on this occasion. And Chef Moore has agreed, and we have invited several people to contribute to the SYNLETT cluster. And we have published already the virtual collection with great contributions. And then we decided to have a web seminar now on this great topic. Today we will have three main talks presented by 8 different speakers. And I would first give a few hints for the web seminar today. So please note that this web seminar will be recorded. And a link of the recording will be sent to all the participants after, shortly after the seminar via email. And this link will expire after 4 weeks. Then second, the audience will be kept on mute during the web seminar. But of course, please feel free to ask your questions in the chat window. And we will try to answer as many questions as possible after the talks. So we would also like to hear your feedback about the WebCheminar, and you can do this, and you can just use the hashtag @Cheminar and tweet at Thieme Chemistry. So in today's WebCheminar, we have, as I said, 3 different talks from 3 main speakers. But the concept is quite new because we have decided that we give visibility to young students and postdocs. And I'm very proud that we have today 8 speakers. Unfortunately, 1 cannot join today. So our plan was to have 9, but today we have 8. But don't worry, we have 3 fantastic talks for the topics. And the first presentation will be given by Robert Göstl and his co-workers Emilia Itzhak-Nau and Davide Campagna from the DWI Leibniz Institute for Interactive Materials in Germany. The second talk will be given by James Mack and Jasmine Crane, and the results from Chennai Sarchan will be presented by James as well. Then last but not least, we will have a talk given by Steve Craig and his co-workers Jafar Wackel and Liki Wang. Yeah, and now I would like really to welcome also our chair of today's event, which is Chef Moore. He's from the University of Illinois, and he gives you now an overview of the topic. And yeah, the floor is yours. I hand over to you now.
Thanks, Steffi. Thank you, everybody, for joining us today. I'll put my screen in presentation mode and get started. Let me go back to the beginning where I really want to begin by thanking Thieme and SYNLETT for being able to think about chemistry in the context of using force to activate and drive chemical change. It's the field of mechanochemistry, and it's a field that has been around almost since the birth of really modern chemistry itself, but had really, I would say, up until recently, been associated with mostly mechanical damage in, say, materials. Then I would say the field looked at biology, where there were many mechanical stimuli that are being sensed in living tissues. And it was evident that we can begin to use force in a much more selective and refined way than we might have thought about from just the context of materials damage. And so today, I think we get to see some of these accomplishments that have taken place over the last, say, 2 decades, where it's been a real shift in the field to begin to think about the selectivity that can be achieved in chemical reactions due to force. I'm really grateful to, again, the organizers today and the Thieme group. I'm also especially grateful to Tom Rovas, who brought this cluster together, the cluster materials together, and the ability to then think about how we might expand this into a broader audience. Let me just begin by introducing a few slides that are going to give you a little bit of background to think about why mechanochemistry has some intuition behind it, and how that intuition connects to chemistry, and ultimately what remains to be done in the field. So on this slide, I've tried to make a connection on the left side between what we think about at the molecular scale, and on the right side, what we think about in our world around us, the macro scale. And so what I've listed on the top are really the kind of measurable quantities that one could associate with an experiment, or either at the molecular level or at the macroscopic level. Let's start on the right-hand side, because these 4 components, stress, toughness, strain, and strain rate, are the measurable characteristics that one might make on a material if that's
Jeff,
what you're studying.
sorry,
Yeah.
may I interrupt you? I think we are not seeing the presenter mode right now. Maybe you can unshare and share again your slides. I think that could help.
I'm sorry, the unshare button doesn't show up. There it is. Okay. Let me try stop. And are you able to see the slides now?
Yes, I think if you can just go to the presenter mode, it should work.
Now I'm in presenter mode. You don't see the slides in presenter mode?
Yes, we don't see them in presenter mode, but I would say at least it's visible. The whole slide is visible, so it's okay to continue, I would say.
I don't see any way I can, I'm seeing it in presenter mode, so that's peculiar. All right, sorry for the technology glitches.
Don't
If you
worry,
can,
continue. It's okay now. It's fine.
okay, I will. And so these are the, on the right-hand side, the kind of measurements that we might be able to make about a material. And they have direct relationships to what we might be able to observe at the molecular level. So for example, stress is the amount of force per unit area that I might apply to this material. Imagining I'm stretching it, I'm pulling it from left to right. And we see an intense color change in the middle, which has been brought about mechanochemical activation. That stress relates to force on the molecular scale. It's hard to define a stress on the molecular scale because we don't really know what it might mean to take that cross-sectional area that when we normalize force per unit area on the macroscopic scale, we can just think about force on the molecular scale. But force alone, this is an important point, force alone isn't really enough to achieve a chemical reaction. There has to be molecular deformation. And in other words, there has to be movement of bonds and atoms. And so on the macroscopic scale, we call that strain. How much have we deformed the material? But on the molecular scale, it might be deformations in torsional changes or bond angle changes or bond stretching that might be involved. The other aspects are toughness, which is really how much energy or how much work we have to do to carry out a reaction. And then there's an attribute of dynamics. How fast are we applying this force? And this is maybe realized in the velocity of the molecular deformation. And so I hope, in this slide really to try to get you to understand that what we do on the macro scale really does have a direct attribute on the molecular scale. What we do on the macro scale and molecular scale do relate, although there are some important differences. And I think the other thing that is really exciting about mechanochemistry is that unlike, say, applying heat to a reaction, which is a scalar quantity everywhere there is a thermal bath and it's basically equilibrating at the temperature we're at, in the mechanochemistry world, we have force, which is a vector quantity. And so we can think about a molecular coordinate and be able to imagine how that force will play out. Finally, I'll just leave you, and this is what you'll see in many of the talks today. The idea is that there's really new functions and new probes that can be achieved. The probes might be sensors of dynamic response, deformation sensors, how much we've moved that material due to force, and what's the magnitude of force that's been applied. We might be interested in functions like detecting damage or how to repair that damage. We might be thinking about things like achieving exceptional performance, or the opposite of what we're talking about up till now, which is how to use chemical energy in order to achieve mechanical force. And so with that, I'm going to end and turn the presentation over to our first speaker and introduce Robert Göstl. Robert Göstl studied chemistry at Humboldt Universität in Berlin, and there he obtained his diploma degree in 2011, and in 2014 received his doctoral degree. Robert went on to do postdoctoral research at Eindhoven University of Technology until 2016, and at that point he joined the DWI Leibniz Institute for Interactive Material, where he began leading his interdisciplinary research group. What I'm really so excited about today is not just hearing from the three of the real pioneers in mechanochemistry, but also hearing about their trainees. And in Robert's case, I think we'll hear both from a PhD student as well as a postdoc. And Robert will kick it off, and I'm very much looking forward to hearing about the latest work that's coming out of your lab, Robert.
Yeah, thank you, Jeff, for the kind introduction. And it's, I mean, it's always an honor to be introduced by one of the pioneers of mechanochemistry. That is very nice, specifically since you shaped the research field that I have now picked up for my independent career. So I assume you can see my screen in full screen mode. If not, then please let me know. Otherwise, I'm just going to go ahead. So I will be talking a little bit, giving a small introduction into oops, now everything is gone again. Try to do it again. So do I still have? I think I still had the controls shared. Okay, so now don't want to take too much time away from Emilia and Davide today, so I'm going to talk a little bit about activating chemical functionality in polymers by mechanical force, and that is twofold. And I'm not going to explain too much in detail about how that works, because Jeff gave an excellent introduction. But I want to go a little bit more, dwell a little bit more on the scale dependent nature of mechanical force, which Jeff excellently introduced already. But I want to add something that I think is very interesting for us. So, I mean, you know that molecules are basically constructed at a nanoscale. Then if you make a polymer out of a monomer, you get a random coil, which is a little bit larger. You put it then into a composite, for example, which is on the microscale. And then in the end, you make a component, for example, a car, right? And if you apply now a macroscopic force to this, your car will yield and brake. You will get a unique mechanical response on all of these scales, for example, crazing on the composite level, chain alignment on the random coil level, and eventually bond scission on the molecular level. And this is exciting for us because it gives us the opportunity to control the mechanical behavior of these materials on each of these scales, and also by mechanochemistry, right? So research in my group comes basically in two flavors. The one thing, on the one hand, we try to understand what happens under force, which means we make optical force probes that visualize force-induced events by changing of optical properties. This in turn helps us to understand materials better. And on the other hand, we try to unleash, let's say, latent functionality, for example, for self-reinforcement, the release of drugs, for sonogenetics, sonopharmacology, and catalysis, and to make, in the end, better materials, right? And so Emilia and Davide will both talk about a little bit more on the right side. Obviously, Emilia will also talk about optical force probes, but both Emilia and Davide will show you how to use mechanochemistry to make better materials. So I'm going to take the time to introduce the optical force probe concept a little bit into you. And this is a field that was shaped by many others before me, but all adhering to this one kind of, let's say, design guideline. So you add a predetermined breaking point to a polymer chain, which cleaves ideally at an increased rate when you apply force to it than the residual bonds of the polymer backbone, and then you activate a functionality. In this case, in our case, it's a reporting molecule. Okay, and we use mainly Diels-Alder adducts of anthracene, and specifically pi-extended anthracene, which are beautiful in themselves, because what they do is these Diels-Alder adducts, they break the pi system of the anthracene. Yeah, so you see here sp3 carbon, and if you activate, or if you pull on them and activate them by force, you regenerate the pi system, giving you a fluorophore with high brightness, which is highly detectable. It's considerably photostable. You can tune the color, and it's quasi-irreversible because of the large activation barrier for the back reaction. And you can connect it to a lot of, or plenty of macromolecular architectures. Yeah, I mean, we've shown this before, that this works in solution, by sonication, in the solid state, so all these standard experiments, which is great, but on the macro scale, this is not where these materials have their strengths, okay? We try to use these optical force probes to solve material problems. I mean, this is what we do. We go in the lab and we're looking for questions that we can answer. And recently we have been able to measure stress concentrations in flow as they are appearing in printing processes, for example, in collaboration with Ronan Daly in Cambridge. We were able to qualitatively map bond scission events in hydrogels, yeah, when they fracture, with confocal microscopy. This is the strength of these fluorophores, that they allow you to do high resolution microscopy. And for example, with Constantino Creton, together we developed the first quantitative approaches to quantify bond scission events during crack propagation here in elastomer networks, also by confocal microscopy. And we continuously go ahead and tailor these optical force probes that they allow us to answer even more questions. For example, we developed these multicolor mechanophores, where your deactivated state or your intact state has a different fluorescence compared to the activated state, which allows you to map in the confocal microscope the non-activated regions of your material in a different channel than the activated material, and then you can make an overlay, and then you can easily quantify directly how many bonds have been broken. Here you see a polyhexylmethacrylate rubber network, basically just cut at this region. Yeah. And these kind of force probes allow us also to locally resolve a particle fracture, for example, in non-uniform materials. So when you look at this at non-activated state, here what you see here is a small movie where we put rubber particles functionalized or cross-linked with this optical force probe into an epoxy matrix, right? So Of course, you can localize your particles, which is great, which sometimes is not easy when you use optically silent optical force probes. But then you activate, when you activate your mechanophore, for example, during crack propagation in such an epoxy, you see that some particles are deformed by crack pinning. So this is what you see here under the bright field microscope. So the crack pins such an elastomer particle. You can localize it easily with the force probe, and you see an activated region. And if you make an overlay out of this, you can, for example, resolve the fracture of an individual particle with subparticle resolution. So you can directly see where the rubber particle here is a micron thick, for example, has been pinched and was broken. Okay, so this is just a small overview, and I don't want to take too much time away from you. And this is why I'm going to introduce now to you Emilia Isak now, who is a postdoc in my lab for, I think now for over more than 2 years. And she's a very international student. Yeah, so she has been all over the place in Europe, in Switzerland. Originally, she's from Poland, and then she graduated in combined graduate programs internationally and all over Europe. And she is the organizational mastermind, I would say, in my lab. She has 2 kids and always manages still to do so much in the lab, takes care of everything, and is truly organizational, much ahead than any of my group. So she will talk a little bit about microgels, so colloidal polymer networks, and how she uses optical force probes to basically identify how they break and then how to improve them afterwards. And Emilia, I think I have enabled the controls for you. I'm now clicking into my slides, and now I'm getting away from the mouse and give you the floor.
So thank you very much, Robert, and welcome everybody. As Robert has just mentioned, in the next few minutes, I will show you how we use mechanofluorophores in order to study mechanical properties of microgels. I will start with a very short explanation what actually microgels are. So microgels are soft materials made of normally spherical microns, and respond to this in temperature, pH, or ionic strength of the environment. They can kind of grow then or collapse, what makes them interesting for a lot of different applications like drug delivery, sensing, or separation and purification technologies. And even though there are a lot of studies concerning the stimuli responsiveness of microgels, there is still a little knowledge about their mechanical properties, what is very important to understand for further processability of the microgels. Thus, we decided to study that using 2 types of microgels. We call them, I don't know why I am moving, but nothing is happening. Unfortunately.
I'll try to click for you.
Maybe, yeah, just click it for me once again. Okay. So we started at using 2 types of microgels. We call them non-dynamic microgels and dynamic microgels. Non-dynamic microgels are those where the polymer chains are linked via strong covalent bonds only, and dynamic microgels are those where we additionally have weak supramolecular bonds in their structure. So we believed that when we apply mechanical force, in the first case, when we destroy the microgels, Robert, could you please just move it? It doesn't work at all. So in the first case, when we destroy the microgels, there will be no way back to the structure they used to have. So Robert, please do it for me. It doesn't work at all. Yeah, you can just click all the time. And then when we have the dynamic microgels, we believed that the weak supramolecular bonds would play a role of sacrificial bonds, so they will dissipate the stress, making the microgels much more resistant to the mechanical disruption. Robert, please click further. Thank you. So we prepared the microgels via precipitation polymerization of vinyl caprolactam. We use mechanofluorophore as the cross-linker, incorporated the mechanofluorophore through methacrylate groups. So again, we have here non-diamond polymer chains. It's really work. Yeah, so we prepared the microgels. We characterized them with dynamic light scattering, and they were pretty monodispersed with size of around 400 nanometers. However, when we had a look at the cryo-TEM, we saw that this microgels had specific core corona structure with hard, highly cross-linked cores and weak soft coronas. Unfortunately, now I see, Robert, the first slide. So yeah, maybe then you can change it. Yeah, once again, please. So we wanted to see whether this microgels still have thermoresponsive properties. So we heated up the sample from 5 to 50 Celsius degrees, and we cooled it down back to 5, and we realized that the volume phase transition temperature of this microgels was around 32, which is in, Robert, could you please just go further? Which is in agreement with lower critical solution temperature of PBCl. So this is the temperature when the polymer loses water and collapses. So we knew that our microgels were still working, and then we wanted to see what would happen when we put them under shear rates, shear forces of different rates and intensities. We started with ultrasonication, so with pretty high shear rates, and how it works here is that the pressure variations induced by the acoustic field cause gradual bubble formation, and when this bubble collapses, this generates solvodynamic shear. So then we took our microgels with different cross-linking degrees into this solvodynamic shearing, and we had a look at the fluorescence intensity and also at size of the microgels. We realized that already after a few seconds of sonication, the fluorescence intensity increased, saying that the mechanofluorophore was getting broken, and meaning that probably the microgels were getting disrupted. This we proved with DLS, so we realized that already after a few seconds of sonication, the size of the microgels dropped, and at some point the microgels started agglomerating. When we had a look at the cryo-TEM before sonication and 20 minutes after sonication, we realized that the microgels totally lost their soft coronas, and the cores started approaching each other through depletion forces mainly, causing the agglomeration which we've seen under DLS. We also realized that with the sonication, we start destroying more and more the highly crossing cores, which were getting more and more fluorescent, what we can see under confocal laser scanning microscopy. Then we wanted to see what would happen when we use of mechanical disperser, for instance. Next slide. With different speed of homogenization. So we see that even with this low shear rates, we were able to destroy the microgels. And this was also dependent on the speed of homogenization. Then we went even down with the shear rates, and we extruded the microgels through needle. And even with this very low shear rates, we were able to destroy the most outer part of the microgels. So then we wanted to see what would happen when we add additionally the weak supramolecular bonds into the structure of the microgel. So here we have mechanofluorophore covalently bonded, and additionally we have catechin bonded via weak hydrogen bonds. So we actually prepared two types of these microgels. Next slide. So those with catechin only and those with catechin and mechanofluorophore of different cross-linking concentration of catechin. And then we realized that the most stable are those microgels with both cross-linkers. Next slide. So we haven't observed any changes in the size of the microgels where we had both of the cross-linkers, and we also didn't see any increase in fluorescence intensity. And the next slide, please. In comparison to those microgels where we didn't have catechin in the system. So it means that the mechanofluorophores were not getting broken, and it also means that the catechin did dissipate the stress, made the microgels much more robust and resistant to the mechanical disruption. And with that, I would like to conclude. So I want to say, next slide, please. So I want to say that we prepared two types of microgels, non-dynamic microgels and dynamic microgels. The non-dynamic microgels consisted of a soft outer corona, this altering the function when we subjected them to very low shear forces. And this all has potential implications for all applications in which shear forces in a solution are expected, including extrusion, injection, and filtration processes, as well as circulation of microgels within bloodstream of living organisms. Subsequently, we proved that physically incorporated catechin rendered the microgels much more robust and resistant to the mechanical disruption, and the microgels with both cross-linkers, so a stable one. Technical problems, ask Davide to continue with his presentation. Thank you.
Okay, thanks, Emilia. It was really hard. Now we see you again. It was a great talk. We had some audio problems, and sometimes with the slide it was very difficult, but you managed perfectly. So we are looking forward now to Davide's talk, and I hand over to you.
Okay, can you hear me properly? It's better if I... Can you hear me? Maybe it's better if I
Yes.
off my camera.
No, it's fine. I think you can.
I can hear you.
We hear you.
Okay. Okay, so yeah, hi everyone. Yeah. Let's see if I can. Hi everyone. I want to give a quick overview about the concept of chemical response upon the application of mechanical force. So I want to report here just a few examples. So chemical response is when you apply mechanical force as output, you get the activation of specific chemical functionalities. I report here some examples. For instance, you can have the activation of cross-linking agent, or also another example, important example, is the activation of some small molecule release, which can be, for instance, the formation of hydroxy groups, or also the generation of acids. Another important example is when the mechanical force is used to break coordinative bond, like in this case here, and this can give rise to the formation of metallic centers or active carbene. And these two molecules, these two activated molecules, can be used, for instance, in the field of mechanocatalysis, because in this case, you can be used these functional groups to as catalyst upon the application of mechanical force. However, another important functional groups which is prominent in organic chemistry and also in organic synthesis and in biochemistry are amines. So amines are pretty ubiquitous functional groups which can be used for different applications, but nothing has been reported so far about the mechanochemical induced activation of amine functional groups, and they are potential for controlling the, for example, signal amplification by controlling the activity of the organocatalysis, or also, for instance, for irreversible reactions, such as the activation of cross-linking involved with the amino epoxy reaction, or also to activate affinity changes within the polymer network when these amines are incorporating into polymer networks. So our idea was to design a specific latent amine when basically the amine is protected with what we can call a mechanoprotecting group. So mechanoprotecting group is something which then is activated by the application of mechanical force, and afterwards you will get your free amine ready to be used for instance, as I said, organocatalytic reaction. However, this is not trivial because there is one important aspect to consider when designing a mechanoprotecting group for amines. The most important is that if you think about, for instance, a standard protecting group for amine, which is a carbamide, you have to consider that if you want to cleave this carbamide group, you have a problem when you want to use mechanical force, because you can see that the cleavage of a carbamide, for instance, is a stepwise process, so an additional elimination process, but the first step, which is the addition, is a slow process, therefore is a redetermining step, and also this process cannot be perturbed by the mechanical force. So you have the problem that the mechanical force in this case cannot influence the overall kinetic of this process. So in order to design such a protecting group, you need to consider that this low step must be the one whose transition state can be perturbed by the mechanical force. So we changed our approach toward the different idea. So we wanted to design a group where you can have an elimination addition process, so the inverse process. In this case, the idea is to use something like this, where you have basically the amine, which is protected with this functional group, which is bearing a weak bond inside the structure, and with this bond you can achieve first the elimination, and then the addition to ultimately give rise to the carbamic acid, and in the end to the free amine. And the idea that we have in terms of what to use as this group was exploiting this oxime-based functional group. These are reported precursor for different kind of radicals, and especially we focus our attention to this oxime carbamate, which is also called carbamoyl oxime. So in this case, you can see that this latent amine here is protecting with this kind of structure, where you have this oxime bond here, which is known to have a low bond dissociation energy. And this structure has also been reported to be also a photo-based generator, because you can cleave this bond, you can cleave this protecting group by application of irradiation and then liberate the amine. So our idea was whether this group can also be used as potential mechanoprotecting group. So the idea that we have and our design was to have such a structure, as you can see again, where you have the latent amine protecting with this carbamoyl oxime group, and then to verify the hypothetical radical mechanisms, either by the application of the light and then also by the application of the force. So the idea was that the oxime bond can be cleaved homolytically in this way, and the intermediate imine radical can then eventually do hydrogen obstruction or dissociation into different pathways. And on the other side, most importantly, this carbamoyl oxy radical is very unstable and normally quickly gives rise to the amine radical, which then upon hydrogen obstruction can then afford the final amine. So this is the structure that we designed, this secondary carbamoyl aldoxime, where this amine here group is a latent amine, which can be eventually used for organocatalysis once it's liberated. So we therefore synthesize our mechanophore. We use this approach using the carbonyl imidazole to make this intermediate, which was then methylated to achieve this good leaving group. And then after a coupling with the proper oxime, we then synthesize our carbamoyl oxime as potential mechanophore. Then we incorporated our mechanophore into two different polymer chain, an hydrophobic Thieme polymer chain and hydrophilic polyhydroxyethylacrylate polymer chain. However, due to limited time, I will just quickly talk about the first one here, the hydrophobic polymer chain. So this linear polymer chain was studied by application of sonication in order to verify the mechanochemical activity of this mechanophore in the middle of the linear polymer chain. To quickly mention the photochemical pathway again, so we verified the photochemical activity of this mechanophore first. So we could prove that the oxime is indeed cleaved photochemically. The amine, the immunir intermediate, can gives as measured product this nitride by releasing of this hydrogen radical, and only traces the aldehyde which comes from the hydrolysis of the imine intermediate. On the other side, most importantly, we verified that this intermediate then gives rise to the amine, which was detected either by chemical derivatization and direct ESI-MS detection. After that, we focus on the mechanochemical activity of our mechanophore incorporated into the polymer chain. So first, we evaluated the activity of the mechanophore by applying sonication to that and comparing it to a reference pristine PMA. And we verified by GPC analysis that there was no breaking of the polymer chain for the reference, but we could observe breaking in the middle of the polymer chain when the mechanophore was incorporating. And so we could assume that something was happening to our mechanophore. After that, so we verified also computationally that this oxime bond can be broken. So we perform a Cogen simulation to understand whether the oxime bond is effectively clipped, and we could verify by the simulation, so by applying the simulated stretching to our mechanophore, that indeed this oxime bond, as you can see here, can be homolytically cleaved. So after verifying this mechanochemical activity of this oxime bond, we evaluated the final structure as well, the product of our mechanophore after application of sonication and irradiation. So we submitted our mechanophore to sonication and irradiation, and we could verify the final formation of this nitride again. As you can see here by the NMR, you can see that there is a merging of this new aromatic pattern here, either by sonication and by irradiation. This A prime and B prime correspond to this aromatic outcome. On the other side, we wanted then to verify the formation of the imine on the other side of the mechanophore, and for that we first perform a qualitative amine labeling. So we applied either irradiation and sonication again to our mechanophore to again create these two products, nitride, which I already show. We analyzed by NMR, and the amine. So the amine was then derivatized using this rhodamine B azothiocyanate, and we created this final product which could be monitored live during the GPC chromatogram with a proper UV channel. So we could see that before sonication and irradiation, there is no UV signal. So the black one, there is no UV signal in the corresponding of the refractive index signal of the polymer for the starting mechanophore. But after sonication and after irradiation, we can see that there is an increasing of the UV signal in the correspondence of the final peaks of the cleaved polymer chain. But you can see that in the shoulders, only the starting shoulder, there is no signal, which means that there is no signal corresponding to the starting mechanophore. So the structure which is not cleaved yet. So after that, in the end, we try to exploit our functionality produced. So the amine which was produced, we try to exploit it for an organocatalytic reaction. So we perform this Conovenagel condensation in situ, ex situ, after either sonication and irradiation of our mechanophore. So what we summarize the result of this experiment, so we could verify, you can see that, so this is a positive control with pure piperidine. You can see that the conversion of this organocatalytic reaction is pretty high, 65% after 2 hours. As negative control, we use sonicated and irradiated pristine PMA, so without the mechanophore in the polymer chain, and we can see that conversion is very low. But on the other side, the sonicated and the irradiated mechanophore give rise to pretty high conversion after 2 hours, which proves that the produced amine after sonication and irradiation is performing organocatalysis. To focus a bit more on the organocatalysis, which is the organocatalysis induced by after sonication, which is the most interesting part. Just to show, we perform a more detailed experiment. So with 3 different trials, you can again see that these 3 control experiment, which are the sonicated pristine PMA without the mechanophore, you can see that the conversion is very low over a reaction time of 4 hours. And on the other side, you can see that when our mechanophore is sonicated for these 3 samples, the conversion is increasing over this 4-hour reaction time. And just to show how the NMR looks like, for example, after 2 hours of these samples, you can easily monitor the conversion by looking at the NMR. So at these D' and C' peaks, which are emerging, which correspond to the scattering product of the phenomenal condensation. And also it's interesting to see that in the same NMR, in this zoom region here, which is this one, you can see that you can also monitor the cleavage of the mechanophore. You can see that this A' and B' here correspond to the produced nitrile. So the aromatic outcome after the sonication treatment. So to conclude, we define a dual mechano and photo base generator, or more precisely, amine generator. So we could bridge photo with mechanochemistry, and we elucidated the process and the mechanism. And you can see we can see the summary of the process in this scheme here for the final for the formation of the final 3 amine. Then we matched the experiment with simulation, and we also could verify this mechanically triggered organocatalysis. So the next step will be the optimization of the mechanophore structure for a better mechanochemical activity, also to try a multi-mechanophore approach in order for a trigger cross-linking experiment, such as epoxy reactions. And also we want to verify the formation of this amine in solid state, which can be useful and interesting for eventual future application within materials. And with this, I conclude my part and give the word to Robert again.
Thank you very much for those presentations. That was very interesting. So in the chat, we would like you to post some questions. We do have some. I'll begin first with the question for David. This is wondering whether or not you've thought about applying the amine release, mechanophore, to mechanically triggered self-immolative polymers. So I think I understand where this question is coming from. I imagine you've already got a catalytic activation by force, so there's some amplification. And if you could depolymerize, there's even greater amplification that is potentially there. I wonder if you've thought about that, or if that's a direction you're planning on moving into.
No, we didn't. I haven't thought about yet, but it's really interesting. Yeah, really interesting input. Definitely something which we can try in the future. Would be a really interesting application. So no, not yet, but this is definitely something that would give even more value to this kind of mechanophore, and would be interesting application. So it's very, yeah.
And then a question came about one of your slides, slide 33, if you can, I'm not sure if you can re-show or not, but you had some sonochemistry, and there were 3 different colors. And how did those 3 different polymers, how were they distinct? I think they were labeled red, orange, and yellow.
I know this is just, so this is just the same, this is just the same polymer, just different, just 2 different samples of the same polymer. Actually, these are just 3 different trials in order to make the result more solid. Just repeated with the same sample in different sonication beds, but it's the same polymer, so with polymethyl acrylate. That was just the repetition of different trials to further prove that the catalysis in the end worked versus the control experiments. But yes, what we also did is, as I also mentioned in one of the previous slides, I also incorporated this mechanophore into hydrophilic polymer chain, polyhydroxyethyl acrylate polymer chain. So I didn't have time to explain also those experiments here, but this is also something that we are working on. So to exploit this amine in water conditions when the force is applied to these hydrophilic polymer chains.
2 more questions, and they're related to being able to look carefully at the chemistry that is induced by mechanical means. One is, did you measure CO2? And the other is there a molecular weight dependence on the behavior that you see?
Yeah, sure. The release of carbon dioxide I didn't measure, because I think there is some concentration issue. So the concentration of the mechanophore is pretty low. So I didn't think about yet a proper analysis for the carbon dioxide detection. Now this is something that I haven't done yet. And on the other question, yes, of course, there is it's dependent on the length of the polymer chain, of course. So yeah, I also performed some kinetic experiment with different length chain of the polymers, and yeah, so the force required is not so low. So the polymer, the length required to break the bond is still pretty high, depending also on the sonication conditions such as solvent concentrations. But yeah, so that's why I'm also working on to try to optimize the structure with structural modification in order to make the bond more labeled toward the application of mechanical force and reduce the force required to break a bond, and therefore also having a lower molecular weight limit in order to break the bond.
And then I have a question about imagining multiple activation of the mechanophore per polymer chain. How is it, as you imagine a solution to thinking about once you break the polymer chain, the ability to transmit force to other mechanophores is lost. Can you talk about ideas that you have, or how do you achieve multiple mechanophore activation?
Yeah, so we are working on the synthesized polymer brushes, so with a certain length of the backbone and then certain length of the arms, where the mechanophore is located at the junction between the backbone of the arms. So the idea we are working on is to break these arms by the sonication and then activate these amine functional groups as side chain on the backbone. So the idea in multi-mechanophores here is to have the arms, so the mechanophore is at the junction backbone arms, and the arms need to have a certain length in order to properly activate the mechanophore and release this amine on the side chains.
Thank you. I am going to end with one question for Amelia, and I was wondering whether or not any the fluorescence, the lack of fluorescence in the case of the dynamic hydrogels, is there any chance that the fluorescence is being quenched in some way by the introduction of the dynamic interactive species?
Unfortunately, I haven't heard the question. I don't know. I'm really sorry about this technical problem. So I don't know if Robert could answer, or if not, if you could just repeat the question once again, please.
Sure. I was wondering whether or not the introduction of in your dynamic hydrogels, if there was the possibility of fluorescent quenching due to the catechol that you introduced.
Yeah, this actually we haven't really checked. Yeah, this is interesting point. But I do not really believe, Robert. What do you think about that?
We can't hear you, Robert.
Okay, yeah, I was force muted. Sorry, people didn't want me talking. So actually, I mean, yeah, we checked. I think we did some cross experiments where we threw catechol together with anthracene when we tried to figure out where the fluorescence is coming from, because the catechene is also fluorescent, which is funnily enough, but has different bands compared to the anthracene. But they don't seem to crosstalk to each other, even though it's due to hydrogen bond formation or something. One could think about this, but with the anthracene, that is not a very good hydrogen bond acceptor, and the concentrations are so low. So we didn't see any. So we couldn't see any interaction between the catechene and the fluorophore.
Well, please join me. This was a set of very exciting results. I'm seeing the, this is unpublished work, and it looks like there's a lot of really exciting and interesting opportunities for really this idea of amplification that can come from a small amount of force, a large chemical change. It really looks very exciting to see the results. So thanks again for really wonderful presentations, Amelia, David. It's good luck to both of you. I'm looking forward to seeing your future work. I'm going to introduce our next speaker. James Mack is a professor of chemistry, and he's interested in environmentally benign chemical reactions. He received his bachelor's degree from Middlebury College in 1995 and went on to earn a PhD at the University of New Hampshire under Glenn Miller, where he studied fullerenes, and went from there to work with Larry Scott, looking at the development of a benchtop synthesis of fullerenes and nanotubes. He joined the faculty at the University of Cincinnati in 2003 and is currently holding the position of Division Dean of Natural Science in the College of Arts and Sciences. I've had the opportunity to see James's career evolve, and it's really a spectacular work. James comes at mechanochemistry from the idea of really using mechanochemistry, as I mentioned, for the ability to achieve environmentally benign chemical reactions. And with that, I'm going to turn the floor over to James, who will have two of his trainees present their results as well. Both of them are PhD candidates. James.
Thanks, Jeff. And so thankful to be a part of this Cheminar with some really big titans in the field. Very, very thankful. One of our speakers won't be with us today because she was sick, so I'm going to present that part of the presentation. So without further ado, so hopefully I'll be able to convince you that mechanochemistry and the way that we do it in this ball milling way is very similar to solution-based chemistry. And there's not a whole lot of difference between the two, but there are some differences between the two, and hopefully I'll illuminate what those differences are. You got to pay attention to temperature and frequency. That becomes part of a process that you need to look at. Sometimes there's mechanistic differences. Unfortunately, I won't be able to go through all the mechanistic differences, but those things are pretty interesting. We use different milling materials, and that can sometimes affect the chemical reactions as well. I'll talk a little bit about that. And overall, we take a lot of the lessons that we learn and apply to a lot of different things. So what you'll see today is a lot of different things that we've learned over the years, and now we moved into biochemistry, and Jasmine's going to tell you a lot more about that. So if you go through and just ask yourself what chemistry is, I like to think of it, there's two things when people ask me about chemistry. There's the cultural side, and then there's the scientific side. And the reason why I bring this up is because when we started this process many years ago, I used to get the statement that, well, that's not real chemistry. And I used to try to figure out what's not real about it. I think we're making bonds and things of that sort. But I think a lot of people are talking about the culture of chemistry, like this is chemistry. You put it in a flask, you move things around, and so on and so forth. If you don't worry about the culture of chemistry, you just worry about the science, creating new carbon-carbon bonds or any kind of bonds that we've seen today. Really, it boils down to, if you believe in collision theory, two particles meet. It doesn't say how they meet or why they meet. If they meet in the proper orientation with the right amount of energy, then you can get a successful collision, or you can get a bond to be created. And so over the years, we've developed ways to make molecules meet. And typically, the way we make molecules meet is we take two reagents, we stick them in a round bottom flask. We add solvent to break down the molecular interactions between molecules so they can freely move and mix together. You may heat it around to get over an activation barrier. Then you remove the liquid, and then you get a product. And it's this liquid aspect that typically is more mass in a reaction than the reagents are. And so typically this auxiliary of a solvent is used. And as you go to chemical processes and scale this up, then this part, the liquid part, becomes a significant aspect, and it could lead to environmental harm. Instead, what we do is we take the same two reagents you would normally take. We stick it inside of a vial with a ball bearing inside, put it inside this mixer mill. The particles are shaken in this mixer mill, broken down to a smaller particle size so they can mix together. Once the chemical reaction is finished, then we get the product out. Sometimes it's the same product, sometimes it's different. It depends all on how you do the chemistry. One of the things when you're doing this is really to discuss the equipment that we use. And so these are reaction vials. We started with custom-made vials in our machine shop, but now there's been commercial vials, and sometimes use a combination of the two. We put them inside this clamp assembly, and then we mill it around in this mixer mill for an extended period of time. Most of the reactions that we use we use stainless steel, but we can make vials out of any material. So you'll see some reactions that use copper and sometimes use nickel, or all sorts of different metal materials. So what's going on in the milling option? So here you can look at a video, and that's pretty much how fast it goes. You can also slow it down, and this is really the ball just going through. So you can imagine molecules are in there and being pulverized by this ball bearing. So there's going to be three topics to discuss today. Energetics is going to be the first one. Catalysis is the second one. And the last one is going to be the mechanochemistry of biomolecules to understand how energetics affects biomolecules. So first I'm going to start with energetics, and that's an important aspect. So when we started, a lot of people was trying to figure out, how does this chemistry work? How much force is being applied to the molecules, and how important is the force? How you measure it? What kind of energetic barriers are you overcoming, and how much of that force is translated to the actual molecules? So we don't have a way of measuring it directly, so we kind of thought of a unique way to try to get at some of this. So we use the model reaction, and the model reaction we use is the Diels-Alder reaction. And we use the Diels-Alder reaction because typically if the molecules meet and there's enough energy, a reaction occurs. If the molecules meet and there's not enough energy, the reaction doesn't occur. So we checked to make sure it wasn't a reversible reaction, so we didn't have to worry about that aspect of it. So all of these are forward-facing reactions. And we wanted to pick a reaction that was somewhere between 30 and 70% in about 3 hours. And so we settled in on this reaction of benzoquinone and 9,10-dimethyl-anthracene that gets you kind of middle of the road. And that way you could go up or down if we change conditions. So you looked at all of those reactions that you saw in the previous slide. And what this point is telling you that at the reaction that was about 9 kilocalories per mole, under all the conditions that we utilized, we were able to achieve quantitative conversion in 3 hours. And these conditions are 15 hertz, either Teflon, stainless steel, or hardened stainless steel that we went through a hardening process. And that allows us to understand the hardening, how important the hardness of the vial is, and the speed of the vial. And so we just basically changed all these conditions. So all 9 conditions provided quantitative yield, and we just kept going to higher activation barriers until we saw something different. So when you go to this activation barrier, we saw the same thing. When you get to roughly around 13 kilocalories per mole, now we started seeing a difference between 15 hertz with a Teflon vial provided no conversion in 3 hours, where if you did it at 21 hertz with a hardened vial, you got quantitative conversion in 3 hours. So, and you got everything in between. So now you can start to see there's some variability here. So then we started to go faster and say, okay, how far can we go? So then we get to roughly around 14.5 kilocalories per mole. You start to see most of these conditions do not give yield. When you get to 15 and beyond, we were not able to do those reactions at all. So it got us to kind of think about the relationship between speed and hardness and force and newtons in a chemical reaction and where this energy comes from. And maybe we thought we didn't have as much energy as we thought we had. And so we started to change the temperature a bit. So as the activation barrier increase, yields goes to 0. And so when we changed the temperature and we made a modification to our milling vial and vessel, then we could start to change how things happen in our reaction system to the point that we could take any point in the middle and make adjustments based on vial speed and vial hardness. So that was really important finding for us, because we thought there's two aspects of this kind of reaction. There's the mixing part, which is how fast things go, and then there's the overcoming the activation barrier part. Sometimes there's the same and sometimes there's different, but it allowed us to basically split the Arrhenius equation so that we can mix really fast and have low energy, sort of like in this system, or we can mix really slow and have high energy, sort of like in this system, and anything in between, and that allows us some variability to occur. So how might you use this information? So we looked at something very, very simple that undergraduates do, which is the reduction of T-butyl cyclohexylone, and looked at the diastereomeric excess between the two. So we looked at it in methanol, what typically undergraduates do, and we looked at mechanochemical conditions to look at the solvent effects that are there. But more importantly, what we're able to observe is basically if you wanted to do something that was diastereomeric or enantiomeric in process, going down to lower temperature allowed you to get better excess. But here we can go down to lower temperature and we could slow down a reaction. And that gives us more control over the reaction than we typically would get in solution. So we were able to find pretty much a new direction to go through. The other thing is that typically when you slow down the reaction conditions, the total reaction is slower. But since we can decouple these two things, now you can look at it that we can go to low temperature, but then we can speed up the reaction by just milling it faster. So we can make the molecules mix together quicker, which is typically something you can't do in solution effectively. Overall, what we notice in these types of conditions are these conditions are typically faster than what you would see in solution. So in this reaction with the an SNAR reaction, looking at what the reaction rates would be at various different temperatures, measuring it over time in a 1 molar DMF solution and looking and doing those comparisons. We looked at a lot of different reactions, and the punchline is overall these conditions are about 9 times faster than what you would see in solution. And that's been pretty consistent. Depending upon a reaction, you can get some changes, but overall it's always faster and you don't suffer any decrease in yields or anything like that for the most part. So that's on the energetic side. So now I'm going to move to the catalysis side of things to show you what you can do on that front. And really what this comes through and the way we think about it, and the way we started thinking about some of these things is entropy. And what does entropy look like in this system? And so when you have a ball milling system or this mechanical chemical version of the system, does a ball come in and smash everything to pieces and everything falls apart? Is that what we mean by entropy in this system? Or does the ball come in like a hammer and nail and force things together? If it forces things together, then maybe we can overcome entropy in this process in a different way than you would typically do in a solvent. And so we started looking at that aspect of it. And so if you look at catalysis and what really happens in a chemical reaction, typically you have a pre-catalyst, and this pre-catalyst is something that's stable, that you can sit on a shelf for a little while at least, but that's not the active catalyst species. That pre-catalyst falls apart to the active catalyst species. That active catalyst species reacts with some reagent to make a complex, then another reagent comes along to make an additional complex, and then that produces the product and you reform your catalyst and you continue the cycle all over again. If you look at some of these pre-catalysts, in this case, palladium tetracus triphenylphosphine, and you looked at the price, you would say, okay, well, these are the components and this is what you would get it for, and this is the pre-catalyst. But if you look at the active catalyst, maybe we could take all of these things together and just look at the raw material. Now you have a reduced cost in terms of the active catalyst because somebody has to put human effort into making this pre-catalyst. But what if the mill can organize all the things that you needed to organize at the same time before you do the reaction? and such that way you don't have to worry about the pre-catalyst aspects. So what we wanted to do was, if we take a piece of palladium and we take triphenylphosphine, that's our reagent A and B, and we use it in a copper vial, could things get mixed together such that while you're putting in the milling reaction, you're organizing everything that you need to organize to create the product out in this way. And so that's what we looked at. We've done this process before using the typical pre-catalysts. So this was results that we've done a while ago. So we used a copper vial as our copper source, our palladium test kit triphenylphosphine, and we milled it for an extended period of time. When we tried it with just the palladium, we didn't get any yield of product, but when we added triphenylphosphine to it and milled it for an extended period of time, we were able to see the product. So this idea seemed that it was working. Then we started looking at the temperature changes. So if you go to low temperature, then you get pretty little or no yield in 3 hours, and we can get to 85% if you run it at 90 degrees. So that's what we did. And so we can do this reaction pretty well, and a lot of substrates we can do by this process. There's also a preference for the iodo over the bromo. So you could go through and take iodo-bromobenzene and this alkyne to do the Schwann-Gaucher reaction, and this is the product that you'll get out in 3 hours. You could take an additional alkyne and add those together. It takes a lot more energy to do this and a lot longer time, but then you could get this bromide out to give you some a mixed product. So we wanted to see if this was just specific to the Schwann-Gaucher reaction. So we looked at Suzuki reaction, and we're also looking at Heck chemistry as well. And I'll tell you, even though the results are not here, it does move forward pretty well in the Heck processes too. So this is the regular pre-catalyst that we typically would use, and that reaction works out really well. If we just use palladium, you don't get this biphenyl. If you add water to the system and heat it up to 90 degrees, then you do get the product that we expected to see. So we're not the first ones to look at the Suzuki reaction in this process. So Suzuki's been done, and I'm going to differentiate mills here for a minute. So we use what's called a vibratory or shaker mill, which is typically a lower amount of energy. Others use what's called a planetary mill. And the difference between the two is typically we use one ball to move things around. And a planetary mill, you use several balls, and you use friction grinding to get reactions to occur. So this is not results done by us. This was done by Göstl a few years back. And in these planetary mills, the rotations per minute is important. And so they would go through and say how many rotations per minute. And these are different balls that they would use to do that. The important part here is you see a difference at about 300 RPMs, where if you do at 270, 250, the reaction doesn't go very well. You need to get to 300 RPMs. Most of these mills do not have a temperature gauge on it, so ours was something that we made in-house, although there's a commercialized mill that has that now. This is a different article by Takis, who was looking at the temperature of these different mills, these shaker versus planetary mills. And this is on the planetary mill side, and you notice that at 280 RPMs, this was done in about 10 minutes. You could see at 280, it gets to about 150 degrees. And so what we did was we wanted to look at 150 degrees to do this chemistry, and we can get this done in about 30 minutes as opposed to 3 hours at 90. So there's a pretty good correlation between the amount of temperature and energy you put in the system and what you can see. And so it's really important to understand that aspect. So on this part of the project, it's really important to understand heat in it and milling speed. Ligands we can use, so some of the reactions we can swap out ligands if we choose. So we've used triphenylphosphine, but we can use different other ligands by just moving things out. So we don't have to get a new catalyst, if you will. We could just take whatever metal we want, another ligand, and just add those two together. In this particular case, the iodo substituent works best, and we could do some chemoselectivity. So that's on the energetics and catalysis, and now I'm turning it over to Jasmine, who's a 5th-year graduate student who came to the Mack group, and she was interested in biochemistry. It's not something that I typically did, but we were really interested in what she was working on. And I'm going to turn it over to her. I'm going to give her control of the mouse and keyboard so she can tell you about the work that she's doing on the mechanochemistry side. So Jasmine, you should have complete control now, I believe.
Okay. Thank you, Dr. Mack. Can you hear me? Can everyone hear me?
Yep.
Okay. I am waiting for control.
You should have it. If not, I could just move it.
Okay, I think I have it now. Yes. Well, no, that you still have it. Okay. Just move to the next slide and I'll see if I can gain control later. Okay. Dr. Mack, can you go to the next slide, please? All right, so yes, thank you, Dr. Mack. So in my particular project, I will be looking at trying to understand how does the mechanical energy within the high-speed ball mill transfer to biomolecules. So as Dr. Mack discussed before, we understand that inside of the high-speed ball mill, the energy that is transferring to the small organic molecules is particularly controlled by heat and frequency. And so there's no way as to right now try to uncouple the two when looking at transformation of organic small molecules. So therefore, we look to employ biomolecules inside of the high-speed ball mill in order to understand how does the mechanical energy transfer to the biomolecules and how does that change their biochemical properties. And looking at proteins under stress has been seen before. Dr. Mack, can you go to the next slide, please? All right, so looking at stress on proteins have been extensively studied in the biological field when it comes to changing the particular structure as well as the functions of these proteins. So the most common types of stress seen on proteins and studied is the oxidation of these proteins due to certain reactions, and as well as denaturing these proteins as well, changing their biochemical functions as well as their native structures. On the more physical side of modifications that we've seen in the biological field is heat denaturation, so allowing heat to denature proteins to either alter their function and as well as their structure, and looking at the changes due to that modification. And what is less understood or what is poorly understood is how mechanical force can change the biochemical properties of proteins, and such that it relates to the onset of selective neurodegenerative diseases such as Alzheimer's and Parkinson's, where the mechanical force within the environment of biomarker proteins can change their function as well as their structure to gain toxic function and to be the onset of these particular diseases and cancers. And so what I'm looking into is not how to cure cancer, but how to understand stress and how proteins respond to stress under mechanochemical conditions. And so the way that we set that up, Dr. Mack, next slide, please. And the way we set that up is we chose a model protein, and that model protein was the RNase T1 protein. And here you see the 3D structure of that protein. And the reason we chose this protein is because it's commercially available and extensive studies have been done on the structure and function of this protein. And just looking at the structure here, you see that the structure is mainly made up of beta sheet structure, which will be important looking at the changes due to the mechanical energy later on. And so the structure also includes a alpha helix structure, which is very prominent in the secondary structure of this protein as well. And as you see here, the function of this protein is also very distinguished in that it only cleaves at the 3' end of the guanosine residue of RNA substrates. And so whenever we look to mechanically stress this protein under different conditions using frequency and temperature, we want to figure out, how the mechanical energy is changing the biochemical properties of the protein. And so looking at the structure piece, Dr. Mack, next slide. So looking at the structure piece solely, we wanted to look at how primary and secondary structure changes due to the mechanical energy or the mechanical force we are imparting on the protein. And so the way we thought to get that information is to use multi-mass spec for primary structure investigation, and as well as circular dichroism to investigate the secondary structure. And so whenever we mill the protein to look at primary structure, next slide. So whenever we milled the protein to look at primary structure, we understood that we were not breaking any amino acid bonds or disrupting any phosphodiester bonds that link the protein and the amino acid sequence. And that was very interesting to the fact that, we are not disrupting the primary structure of this protein. The initial idea was once we add the mechanical energy to the protein, all things would potentially be disrupted. But here we see whenever we introduce mechanical energy at different frequencies, and I started at a very low frequency of 8 Hertz and increased the frequency to 23 Hertz, val Hertz, and we see that there are no significant changes in the primary structure. But whenever we look at secondary structure changes to protein at the different milling frequencies, we do see changes. Dr. Mack, can you go to the next slide? And here in this graph, the first graph that you see is the circular dichroism graph of the secondary structure of the protein once it has been milled. And just walking you through, the blue line that you see here is the non-mill standard protein. So like I introduced before, I shown before, the protein is majority beta sheet. And so the blue line is the representation of a native beta sheet protein. And so once that protein was milled at higher frequencies, you see that that beta sheet structure starts to decrease, and that we are implementing an alternative conformation to this protein. And it's very important to understand that we are seeing these changes solely due, or majorly due to the mechanical energy inside of the system. And we wanted to identify this by doing a denaturation study of the same protein. And this is the graph that you see next to the mechanical energy studies. And so whenever we did this, we wanted to ensure that the changes that we see are solely due to the mechanical energy inside the system and not just due to the thermal energy that we dissipate inside the system. And you see that the profiles are significantly different, and that secondary structure due to milling is not all decreased as you see whenever we thermally heat this protein. So with this, we understand that we're not totally disrupting the secondary structure of the protein, but we are making small and incremental changes with the conditions that we're using, but not big incremental changes where we decrease all secondary structure, as you see with the thermal denaturation of that protein. And so the next steps that we wanted to understand is how does the structure changes relate to the function of the protein? And next slide, please. Yes. And so the way we thought to do this is to use multi-mass spec again to identify digestion products using the protein in a enzymatic assay. And so the whole idea was to try to understand how we are affecting the protein function as we see, secondary structure changes whenever we mill the protein. And so we used we built our own synthetic RNA substrate for the protein to be incubated in the assay. And so with this particular RNA substrate, we understand that there are theoretical digestion products that the protein should produce based on its functionality. And we went to look at the appearance or disappearance of those particular digestion fragments whenever we ball milled the protein at the frequencies that we used. And so here in the table, you see the predicted digestion fragments that we should see for the native or regular function of the protein. And then you also see the synthetic RNA oligo that we should see the cleavages at every G residue. So here in the RNA sequence, I have color-coded each of the fragments. So, and then I'll tell you why I did that a little bit later, but each of the fragments here are color-coded, and those are the fragments that we expect whenever we do the enzymatic digestion with the ball mill protein. So the results that we had, next slide, please. Thank you. And the results that we had showed very interesting here. So just walking you through the table, we chose a unmilled standard protein just to make sure that we get all theoretical digestion products that we expect, just to ensure that our conditions were optimal. And as you see, we do get every single predicted digestion product there. Whenever we start to add mechanical energy at 8 Hertz, we still get our same digestion products that we predict. So we understand milling the protein at 8 Hertz Val frequency, we still get a relatively good function still of the protein. But more significantly, whenever we milled at 11 Hertz higher, we lose the digestion fragment that is in the center of the sequence. So once we mill at 11 Hertz and higher, we see the disappearance of the cleavage of that digestion product. And so that was giving us a little information of how the cleavage pattern of the T1 changes after ball milling at 11 Hertz and higher. Going on, milling at 12 Hertz and higher, we also see the disappearance or the decreased cleavage of the next two sequences or the next two fragments that are also in the center of the sequence. And that also decreases over time. But what stays are the cleavages on the sides or on the ends of the sequence. And we have an idea of why the protein continues to cleave, and is due to the fact that looking at the secondary structure, we're not totally disrupting the entire beta sheet of the protein. So therefore, it still should have some function, and we see that it does have it has function at cleaving the ends of the sequence, but it does have trouble cleaving the center of the sequence. And to try to further get an understanding of the T1 cleavage pattern after ball milling, we also looked for any miscleavages that the T1 may have had and see if there were any common miscleavages that we identified in the enzymatic digestion assay after ball milling the protein. And what was significant was that, yes, we did have common miscleavages in ball milling the T1 at the 11 hertz frequency and on, but we did not identify any miscleavages in the unmilled protein or the protein milled at 8 hertz. And these miscleavages are significant in that they're located on the ends of the sequence as well. And so we understand that there is a structure to the sequence that is probably difficult to get to due to the changing of the T1 secondary structure. And so we want to understand how or, where are we affecting the protein to where we see miscleavages and cleavages only at the ends of the sequence. And so, next slide, Dr. Mack. And so we wanted to take a closer look at the active site of the RNase T1. And so the active site is mainly in the beta sheet region of the T1. And so that gives us information about the changes in the cleavage pattern. And next slide. And the residues, yes, and the residues that are located within that particular pocket are the histidine 92 and 40 that are a part of the catalytic mechanism. And then there's also the other residues that are a part of the phosphate stabilization. And so the fact that those are located within the secondary, within the beta sheet pocket, we understand that we could be affecting those to where we see these miscleavages and the disappearance of certain digested fragment cleavages as well. More specifically, next slide, Dr. Mack. Yes, okay, thank you. More specifically, we think that we may be affecting the B2 site. And just to give you a little bit of information, the P site is, so most proteins have P sites and B sites. The P site is the catalytic and the phosphate group recognition residues that I shown before. And the B site is the base recognition of the 5' and 3' ends of the residues. And we see that we have good base recognition with pyrimidines, which is specific to the protein, but we do not have any base recognition for purines. And so that may be due to us disrupting or deforming the B2 site, which is the 3' base recognition site, to where we are distinguishing between the purines versus pyrimidines. And so we will have to do more studies with that to understand if we are affecting this B2 site to where it does not cleave at purines, but it does cleave at pyrimidines. And then we could understand just a little bit more how the mechanical energy from the ball mill is affecting the active site or the catalytic site of the protein, which gives us our miscleavages, as well as the loss of digested fragments. Next slide, Dr. Mack. Okay, thank you. And so just to summarize and conclude what we understand thus far about mechanical energy transforming to biomolecules is that primary structure does not show any disruption due to mechanical milling, but secondary structure does change, but that change is not as significant when denaturing the protein. Milling at 12 hertz or higher does cause activity and specificity changes, but activity does remain whenever we do mill it at our highest frequency. So that still gives us an idea that this protein is still functioning. We see most miscleavages at the pyrimidine site. So most of the adenine and guanosine sites have miscleavages, but we don't see any miscleavages at the purine site of the sequence. So we may be affecting the base recognition, the B2 site base recognition of the 3' end of the sequence to where it does not cleave at those particular sequences or residues. And we would need to do more studies, possibly with other sequences, to get a better understanding of the changes in the cleavage pattern of the T1 after ball milling. And so that's what I have for biochemistry inside of biochemistry along with mechanochemistry. And I will turn it back over to Dr. Mack to conclude.
Thanks, Jazz. The only thing that I have left is acknowledgments for all the collaborators and students that's worked on this project over the years, and be happy to take any questions.
Thank you so much for those wonderful presentations, Jasmine and James. I think it's really exciting to see how the mechanochemistry is really hitting new directions, and especially the work that you showed with the T1 RNAs. We do have a couple of questions coming in. First of all, Jasmine, I wondered if you were after mechanical, after you do the ball milling, is it possible to recover the secondary or the secondary structure that's lost? You showed the CD, but I guess the CD, were they recorded in aqueous media? Is that right?
Yes, yes, yes. So the protein is milled in a solution, and then after milling the protein, the CD is taken right after. So preparation is done for CD, of course, and then right after the preparation, CD is taken.
So that's an irreversible change then. It seems like you never see a recovery of the CD signature.
Yeah, so up to a certain point, correct. So we have done heating trials as along with the milling trials, and we see once we pass a threshold of mechanical energy, the protein does start to change back to its original structure. So it does try to fight back to go to that lower energy state. But that's once we pass that certain threshold, and we're still doing trials right now to see what exactly is that threshold. Because yeah, when I milled them, I automatically took the CD. What I have not done is taken the CD 2 or 3 days later to see if that structure pattern changes. But I will look to do that soon. Yes.
Thank you. The next question is for you, James. Is there limitations in terms of reagents that you can use in ball milling? And in particular, one of our participants asked, for example, is it possible to use shock-sensitive reagents like azids, for example, under ball milling conditions?
Let me unmute myself. There's a lot of variation. So the one thing that we have some difficulty using is a gas. So if you think about mixing things together and having a ball come and kind of be the facilitator, if it's a gas, the gas can kind of move out of the way. And so there's been a number of publications that have been seen where you can do some of these reactions, and the ones that I showed, and I didn't mention this, were all done in ambient temperature, ambient air. So vaporous water and oxygen, since most of the time there's the oxygen or water dissolved in a solvent, because solvents typically is a Trojan horse for those types of things. When you don't have that around, then the chemistry works out really well because the vaporous oxygen and water is moved out of the way, so the molecules don't even know it's around. If you wanted to use a gas in some way, shape, or form, then it causes a problem because you can't hit it. So that's on the one front. In terms of shock sensitive materials, you can use shock sensitive materials. So we did click chemistry where we did a one pot reaction where you use sodium azide or any azide. Used a copper vial as the catalyst to do the chemistry, and then you had an alkyne. So you take an azide, the alkyne, and the copper vial, you mill it together in about 15 minutes, you get your triazole. We've never seen any evidence of explosion or anything like that. And I think one of the things that Jasmine demonstrated was the fact that depending upon how fast you mill these things, there's not a whole lot of energy that comes through to really cause those types of reactions to occur. So it's really controllable in that way, such that when we started this biomolecule project, most of the biochemists said, there's no way the protein's gonna survive. And you can see you can mill it for extended periods of time. So I think she milled it for an hour. And the protein does something different than it did before, but it survives the milling process. So the energetics that are there are not as high as one might think. Now, I don't know if it's the actual energetics or how much energy is actually transferred to the molecule versus the vial in other places, but the molecules pretty much hold up.
Fascinating. Another question for James. The Suzuki-Mayara reactions that you showed were with boronic acids. The participant asks whether trifluoroborates would be another possible substrate.
We believe so. We think those will go even better. We haven't tried that yet. So right now, what our thought process is really trying to understand this entropic aspect of palladium and what mix and match. So our idea is having a toolkit, let's say. You have a metal catalyzed reaction, so in this case, palladium. And our ultimate goal is to have a polymer supported ligand such that you can recover both at the same time, so you can mix and match whatever you want. That's just where we're going, but we think different variations would work as well. We just haven't tried it yet.
And then this question is for Jasmine. I personally was fascinated by this idea of the link between mechanical environment and mechanical forces on disease pathology. Has that been very clearly elucidated? Is there strong correlation evidence for that, or is this a hypothesis that you're pursuing and trying to test?
So it is a hypothesis. So I read many review papers about looking at mechanical stress on proteins. It has been highly hypothesized how much mechanical stress that the significant proteins can undergo before they gain toxic function in selective cancers and diseases, but how the mechanism in which these proteins change and how these proteins structure changes as well is not clearly understood. And so right now, I guess the information that I get from my project can help in the understanding of the mechanism in which these proteins undergo, mechanical energy, and they've changed their structure and function to, a different modification than to where it was at the beginning. And so, yeah, more information would definitely need to be understood to get a clear hypothesis about how the changes to proteins affect disease and cancers and everything. But I think we're starting to just get on the brink of it, the brink of understanding. Yeah.
Well, thank you very much for 2 great presentations and really moving the field in exciting new directions. So with that, before our final set of presentations, I'm going to turn the floor back over to Stephanie, who I think is going to give us an announcement.
Yes, so yeah, thanks to all speakers and also Jeff to you for a great moderation. Yeah, I think all of us are already looking forward to the final talk. But before, I really would like to give you an overview of our products and also an outlook of our next webcaminars and also an outlook of our next upcoming topics we publish in our journals. Yeah, so I would like to share my screen again. Yeah, Thieme Chemistry. I think you might know other products from this publisher. So Science of Synthesis, the database, or pharmaceutical substances. And yeah, we publish journals like Synthesis, the full paper and review journal, SYNLETT, and SYNFACTS, and Organic Materials, which is an open access journal focusing on organic materials and applications. And yeah, the web seminar today was initiated by a cluster published in SYNLETT. Tom Rowes was the editor of this cluster, and Jeff Moore, the guest editor. And we have published several contributions so far, and I would highly recommend the articles to you. And there's still a chance to submit papers for this topic. We keep this collection ongoing for a while. So if you are interested, yeah, please submit your article to SYNLETT. We have several upcoming topics. So we have special topics in synthesis. One is on phosphorus redox. Another one on arcane chemistry and synthesis. And we have several clusters coming this year also. One is on organic photoredox catalysis, another one on dispersion effects, and a third one is on machine learning and artificial intelligence. So yeah, consider also the journals if you are interested to submit papers in this field. In organic materials, we currently publish 2 focus sections on supramolecular chemistry and another one on porous organic materials. Yeah, and I would like to also invite you to join our next upcoming WebCheminars. The first one is celebrating organic synthesis in Latin America, where Gary Molander is chairing the event. And we have 3 fantastic speakers also, and the date is April the 27th. And yeah, last but not least, another journal-related special WebCheminar is coming on asymmetric CH functionalization, takes place on May 11th. Our synthesis editor, Professor Liu-Shu Gong and Professor Gong Chen will chair this event. And we have, again, 4 fantastic speakers. And I would also, yeah, welcome you for this web seminar as well. So I think now it's time to continue with the final talk. And I would like to hand over to Jeff again.
Thanks, Steffi. It's my pleasure to continue to show some of the work that is at the forefront of mechanochemistry. And we have with us Professor Steve Craig from Duke. And Steve will be joined by 2 of his current PhD students. Steve received his PhD from Stanford. I think I would classify Steve as being trained as a classical physical organic chemist. He had 2 stints after that, and before he joined Duke. One was at DuPont, and he was also a postdoc at Scripps. I've known Steve for a long time. He really has been a trailblazer in the field of polymer mechanochemistry. He's currently directing the Center for Chemistry of Molecularly Optimized Networks at Duke. And he's going to present, along with his 2 students today, some of their most recent work. Steve, the floor is yours.
Wonderful. Thank you, Jeff, very much for the introduction. Thanks to Thieme for putting together such a wonderful WebCheminar. And I'm yeah, delighted to join with people whose work continues to influence the way that we think about mechanochemistry and the potential of the field, to share some of the things that have been going on in our group here in the last couple of years. And I'd like to begin by echoing the way that Jeff started this symposium, right? Which is just to remind you that essentially when you say stretch a polymeric material, what's happening macroscopically is also happening microscopically and molecularly. Right? So that as I take this macroscopic material and stretch it, I'm stretching molecular components. And the nature of that molecular response is exactly what you would imagine that it would be, right? That you've got polymer chains that undergo conformational changes. You start to, as you stretch further, to form the bonds, bond lengths, bond angles between atoms. And ultimately, it culminates in a chemical reaction. Historically, the scission of bonds that often is correlated with the fracture and breaking of the macroscopic material. And so I thought James made this point really well, right? That if often you look at manipulating mechanically macroscopic materials and you think of that as just a purely mechanical or physical process, but fundamentally it's chemistry. And so we really like to think about this deformed, strained network as being its own kind of chemical reactor that's undergoing exactly the same kinds of molecular responses that are at the heart and soul of the introductory organic chemistry course that all of us took many, many years ago. And so we're really motivated still by a couple of fundamental questions, right? Can I take that chemical potential of this physical reactor, if you will, and use it to direct other kinds of chemical outcomes? Or could I use the chemical outcomes and use that to influence material behaviors? And we'll give a couple examples that today that reflect, I think, both sides of that mechanochemical coin. And so one of the things that's great about this field is all the ways that you can imagine pursuing interesting questions. And some of the questions that motivate us that we'll touch on today involve essentially taking some of the color changes that you see in this movie here, and Robert and his team showed wonderful examples of sensing mechanochemical feedback through color change or spectroscopic changes today, and use it to ask and perhaps answer questions, right, about how materials are breaking down. Can we change the physical limits of materials today, or can we, instead of generating a color, can we, as Robert's team talked about, I think Davida at the end of the talk today, can we convert those mechanical cues into robust chemical triggers? All of these questions for us begin with asking about fundamental questions about chemical or really mechanochemical reaction development. And one of the reasons I'm really excited to be talking before this Thieme Symposium WebCheminar today is because I know this community is really interested in chemical reaction development. And so we'd like to talk a little bit about that and begin with a story that echoes some of the mechano-base story that you saw earlier today about taking mechanical forces and converting them into other kinds of chemical triggers. And if you think about potentially useful chemical triggers, certainly bases are one. I might argue that pound for pound, the proton is still the most versatile chemical reagent that we have. And so you can imagine all sorts of acid catalyzed processes that might have an influence on material properties. Protons are very small, and so they hop around fairly quickly. And this sort of begs now a reactivity challenge, right? How do I mechanically release a strong acid into a material environment? Not surprisingly, I think this has been a motivating question for a number of folks, and we were inspired by work that Jeff and his group have done over the years, reporting a couple of other mechano acids. But that we saw as having some limitations, at least in the context of the kinds of questions that we were ultimately interested in applying these to. And so we were motivated to come up with mechanically triggered acid release that would be synthetically really accessible, would give thermally stable latent mechanoacids that had good activity, and importantly, that could be generated without the breaking of a polymer chain, right? Which would essentially end the activation event. So could we get the release of many copies of an acid across a polymer chain under a high enough force of tension? And the path to this really stemmed out of a mechanistic insight. And this was based off of some mechanochemistry that my group reported well over a decade now, which is the mechanically coupled ring opening of dichlorocyclopropanes. And the key point here first is that what I've shown here is a polymeric repeat of this mechanophore. And so there are actually many copies of this mechanophore potentially along a polymer backbone. And its activation doesn't break that polymer chain, but instead allows you now to essentially completely remodel the polymer backbone under a high enough force of tension. We will, throughout the talk today, both I, Li-Chi, and Jaffer, show pictures of a single mechanophore, but in the vast majority of cases, we're really looking at many copies along the polymer backbone, and so that will be implicit in the structures that we show. The mechanism of this electrocyclic ring opening essentially involves classically a lot of allyl cationic-like character along the backbone that accompanies the migration of the chloride from one carbon to its neighbor. And so Yangju Lin, who graduated, as you can see, during the pandemic, looked at this and thought, well, what if I put a pi donating substituent, like an alkoxy group, onto that mechanophore? Surely that would enhance the reactivity. And in fact, using some kinetic measurements that we won't talk about today, Yangju was able to show that you could increase the rate of that chemical reaction essentially by 1,000,000-fold at sort of the forces relevant for activation. And this substituent effect came with a bonus, essentially. And that's that if you look back through the old literature, Yangju discovered that alkoxy substituted cyclopropanes not just were easier to open thermally, but also were accompanied by HCl, spontaneous HCl elimination. And so we asked the question, if I stretch a polymer chain bearing these putative mechanophores, in this case, using pulsed ultrasonication in a THF solution, would that trigger the release of HCl? And so he tested this by adding essentially a pH indicator, a rhodamine dye, into the solution. And what this picture tells you is that that idea works really well. Okay, so you can mechanically trigger the release of HCl. Because there are multiple copies along the backbone, you can trigger the release of of around 60 or 70 equivalents per polymer chain breaking. And I want to point out here that as exciting as this was, it was far from perfect. In fact, there are a variety of reaction outcomes here, not all of which actually produce HCl, but at least when you do the mass balance, indicate that we've also lost some methyl chloride in addition to HCl chloride in at least some of the pathways. Yeah. Okay, so very excited about this. And then it raised questions about, could we extend this to other triggers? And fluoride is another great trigger, especially in the context of material remodeling. And so this was a question that we were wondering about. Right? So we've shown that these alkoxy substituted dichlorocyclopropanes give reasonably facile release of HCl. Could a similar analogous structure with difluorocyclopropanes also be equally facile when releasing HF? Now, there are a lot of reasons why you might not expect this to be the case. Nonetheless, it was a challenge that Li-Chi took on, and I will turn this now over to her to allow her to tell you about just a part of the work that she successfully defended. So 3 cheers for Li-Chi at her PhD defense just a couple of weeks ago. Okay, so Li-Chi.
Oh, thanks, Steve. So will you move the slides for me, or should I try?
I think I've given control to you. If that turns into a problem, let me know.
Okay, sounds good. Hi everyone. So today I'd like to share you the story of mechanically release of hydrogen fluoride. So despite the challenges that Steve just mentioned of why this system can potentially fail, but we still have to start from somewhere, right? Because of the previous success of mechanically release hydrogen chloride, we think this methoxy-GDFC is still our best shot and a good starting point. So we went ahead and put our effort to synthesize such molecule. And we first tested its thermal stability. And such molecule is actually showing a great thermal stability. There was no ring opening reaction after heating at 140 degrees Celsius for after 1 day. And even at 160 degrees Celsius, it slowly ring opens over the time scale of hours. Steve, can you click? So as expected, this fluoride version is just less intrinsically reactive compared to its chloride analog. But how about mechanically, right? So we apply mechanical force on this methoxy-GDFC molecule with the ultrasonication technique. And so in order to visualize and detect the generation of hydrogen fluoride, we also use this silylated coumarin probe that selectively detects the generation of fluoride. And as you can see here, under ambient light, it shows a pale yellow color of after the sonication. And under UV, it's even more obvious. It shows a very strong greenish blue fluorescence. So both color changes here indicates that we are actually generating the hydrogen fluoride after sonication. But we really want to dive into the details of what are the actual reactions and what are the products that we have formed. So, and I also want to mention that we added excess amount of triethylamine during the sonication to trap the hydrogen fluoride generated within the solution because of the safety concern, So we took a bunch of mass spectra to characterize the product that are generated. And as you can see here on the fluorine mass spectroscopy, there are 2 new peaks generated after sonication, and with an integration ratio of 1:1 all the time. And so one of them represents the triethylamine hydrogen fluoride complexes, and the other is the fluoroalkane on-chain product. And it's worth to notice here that this reaction is actually much cleaner compared to the release of hydrogen chloride. So if you still recall from Steve's previous presentation, that hydrogen chloride are actually forming 3 different on-chain products, whereas the fluoride version only has 1 exclusive on-chain product. So next slide. So now let's talk about some numbers. So despite the thermal stability, the fluoride has a much higher thermal stability compared to the chloride version. But mechanically, at the same force, their ring opening rate constants are very similar to each other. So that means they are very similar in high force reactivities. Next slide, please. So we are wondering why, what causes these differences. So our collaborator, Ilya from MIT, calculated this mechanism for us. So under force, we found out that it first ring opens the methoxy-GDFC 3-member ring and forms this diradical species, and then goes through a fluoride dissociation. And now this dissociated fluoride wants to bind back with the molecule, and so it has 2 choices. But energetically, we found out that forming the quaternary carbon is more dynamically favored. So everything is smooth so far. But the problem is when we try to calculate this from this intermediate to the final product, we cannot find a pathway that are energetically reasonable. Like all of them are super high in terms of the energy, which cannot be possible during the ultrasonication condition. So it must be something else. So we reconsider the possibility of the triethylamine playing around in this reaction pathway. So it turns out the triethylamine can trap this diradical species and then forms a transition state. Next slide, please. That are shown here. And so with this transition state, first is very energetically reasonable compared to the other pathways. And also it can explain the experimentally observed regional selectivity of the actual product that we are observing. And also this pathway is very unique for the release of hydrogen fluoride compared to the hydrogen chloride. So now that we have demonstrated the efficient release of hydrogen fluoride mechanically and also explained its mechanism, we really want to apply it to something. So as we know that hydrogen fluoride selectively cleaves the silicon-oxygen bond. So the idea here is that we want to synthesize a polymer that contains both the HF releasing group and the HF cleavable group. So under sonication, the HF releasing group will release hydrogen fluoride, and then it will kicks off the silicon-oxygen bond and triggers polymer degradation. So we go ahead and synthesize this polymer that basically contains three parts. First is the HF generating part, and second is the HF cleavable part, the silicon-oxygen part, and the third one is just a non-bonding copolymer. And we also synthesize a control polymer that basically has the similar components, but just without the HF generating part. And then we sonicated both solution under similar conditions and compared these molecular weight changes. And as you can see here in the GPC analysis, even only with 3% HF generating part, it can significantly degrade the polymer faster compared to the control polymer. So that basically ends the story of hydrogen fluoride release. And now I'd like to move on to another story that is about the mechanical activation of cubing. And the story starts with this cyclobutane molecule shown here. So the mechanochemistry of cyclobutane has been extensively studied by many groups. So if you pull on the 1,2 geometry, it will break in the middle and form two double bonds. So we wonder about this cubing molecule because it has a lot of cyclobutanes here, and also the ring string energy is very high, is of 162 kcal per mole compared to 26 kcal per mole in the conventional cyclobutanes. So we wonder if we pull on this cubing molecule, will it break? And if so, what are the products and pathways? So we put our effort to synthesize this cubing containing polymer. So after sonication, you can see on the RMR spectroscopy that two things here. So first is the decrease of peak intensity in the cubing protons that are around 4 ppm. And second thing is that there are a bunch of new peaks showing up. And after careful analysis, we think we assign these protons to the same tricycle octadiene as the mechanical activation product. And it's an efficient conversion and also just forms this one single product. And as you may have already noticed that actually there are a bunch of ways that you can pull on this cubing molecules. So namely 1, 2, 1, 3, and 1, 4 positions. So despite the fact that all of these molecules can be thermally activated and form this cyclooctatetraene as its product, mechanically, only this 1, 2 cubing can be converted to the tricyclooctadiene product, whereas the other two Pauling geometry, the 1, 3 and 1, 4, does not activate it. And in terms of the force reactivity, it is much more reactive compared to the conventional cyclobutane. It's 10 to the 4th more reactive. And And also, it should be noticed that this tricyclooctadiene is a mechanical stable product. It will not further break to the other two molecules. And this is because of two reasons. For one, this formed product are anti-aromatic, which is much more unstable compared to the syn tricyclooctadiene. And second is the pooling geometry is not as efficient as the most efficient 1,2 pooling geometry. And now I will hand it over to Jeffrey, who will talk about how we can use these types of reactions to really explore some opportunities and applications into bulk materials.
Great, fantastic. Thank you so much, Lichi. I will now talk about the effects of molecular substituents and how that they can affect the mechanical properties of a polymeric material called a gel. So the molecule that I've shown here has 4 chains of polyethylene glycol, each terminated with an azide. I'll refer to this as a PEG star. And this compound can undergo a quack addition with this dialkyne linker to link the ends of one PEG star to another PEG star. We briefly spoke about cyclobutane being mechanically active. And here we perform this quack addition with these dialkyne linkers, 2 of which contain these mechanically weak cyclobutane compounds. When we end link these PEG stars together, we build a polymer network. And when that network is swollen in a solvent, it's referred to as a gel. And the idea is that the cyclobutane linker in gel 1 is very weak, in gel 2, relatively weak, and in gel 3, it's relatively strong. And so these gels are made under identical concentrations and conditions. The only difference between them being the identity of the cross-linker used to connect the PEG stars together. And we can compare the physical properties of these gels. The graphs shown here show how much energy is required to distort the materials or how much solvent the materials can absorb. And as you can see, the values are virtually identical since the gels themselves are very similar. But when we test the energy required to tear the gels, what we find is that the gels containing weaker linkers actually break much more easily than the gels with stronger linkers. And per polymer chain, there's only one of those weaker strong linkers, but the presence of those linkers still greatly influences the macroscopic properties of the material. Cyclobutane is mechanically weak and undergoes a force-mediated 2+2 cycloreversion to break in half. In the case of the very weak linker, what ends up happening is that the diaryl substituents stabilize the diradical transition intermediate and facilitate the cycloreversion. And while we did make the materials easier to break in this case, we were able to leverage this chemistry to build materials that grow longer, absorb more energy, and show improved tear resistance in later work. I'm going to push forward with this. Great. I'm going to attempt to play this video, but Steve, if you could take the wheel and attempt to play the video for me, that would be great. Yeah, and we've talked about synthesizing and characterizing mechanoacids and polymer gels, and we can use chemical design principles to combine and target properties within different materials. And here we have a hydrogel containing methoxy-GDCC, and it releases acid upon mechanical trigger and still recovers its shape even after harsh strains. And I really like this example because I love demonstrations done with a hammer, and it also combines some of the chemistry and the different themes that we've spoken about today into one material shown here. And one last thing I'd like to mention is that we don't often consider materials from a chemical perspective, but materials such as tires are fundamentally very large macromolecules. And in the United States alone, millions of tires are landfilled annually. Those tires are comprised of polymer networks, and the fracture of those tires is inherently chemical. Identifying ways to increase the lifespan of those tires or program in recyclability within those materials will require strategies that take advantage of the inherent chemical reactivity within those polymer networks. And we hope to contribute to those strategies with our present and future work within MONET, our NSF Center for the Chemistry of Molecularly Optimized Networks. This work is made possible by our funding from MONET, our collaborators, and our lab. And one last thing I'd like to say is just to thank the audience for their time and for listening to us. So thank you. And with that, I think we would love to take any questions that you might have about any of the work we presented today.
Thank you, Jaffer and Liche and Steve. There are questions that are coming in. Let's begin with, we go back to the beginning. In the HF release, the HF releasing mechanophore, is it possible that you drew a mechanism where there was homolytic cleavage? The participant asks whether or not it's possible, or have you considered heterolytic cleavage?
Yeah, so you want to take that, or should I take that?
Yeah, I mean, I think we could say a couple. So yeah, short answer is yes, we've considered it. Computationally, what comes out is that there's this really interesting intersection between the two. That it essentially starts out as homolytic, and then kind of hops over through an internal single electron transfer to a more dissociative ionic character. So like with any mechanism, I think the reality is often a little bit more complicated than the way we actually draw the draw the arrows and think about it. And this is no exception. We on related systems, we have done things like to use essentially radical traps to intercept those kinds of intermediates along the way. Haven't done it for this one specifically, but for other difluorocyclopropanes we have. It's a great question. Thank you.
This question's for Liche. Is there a scientific explanation for the selectivity of the cubanes 1,2 compared to 1,3 and 1,4 isomers?
Yeah, that's a great question. So our short answer to this is that like 1,2 is just the most efficient Pauling geometry, like in the cyclobutanes, and we think the 1,3 and 1,4 is not as efficient as the 1,2, and maybe the force is more dispersed through the different bonds, and so not big enough to open that cubing.
And then
yeah, maybe I'll just add to this.
Go ahead.
Yeah, sort of a fun. One of the things that I like about this field very much is kind of how physical and visual it is. And if you like go and start building, introductory organic model kits with these mechanophores and just pulling on them with your fingers, you can start to appreciate the ways in which these forces couple on to potential reactivity. Sometimes it's not that obvious, but often, and I think in this case it is.
Jeff, this one's for you. When we work with the star pegs and generally find that the regular network structure leads to extreme breaking strains, you showed that the elastic moduli vary depending upon the identity of the group attached to the cyclobutane ring. And I think the question is, do the breaking strains track in a similar way?
Yes, so I believe that the point of the graph of the modules was actually that they're pretty consistent across the different shells, but that the tearing energy is lower for the very weak linker and then increases as the strength of the dialkyne linker increases.
Right, thank you. And then for the ladder generated after the cubane activation, could it be used for a secondary reaction? Like, have you thought about a Diels-Alder reaction?
Yeah, absolutely. So I think we can potentially use this formed product as some cross-linker and generate some other useful applications.
Another question.
To hear suggestions. Yeah, so please, always send us a note and let us know your best ideas.
And then we have a mechanistic question about the cubane opening. The participant asks whether it's, or assumes it's a stepwise opening, not a concerted opening, isn't it? And then, so I think the question really is, what do you know about how those two bonds break in asynchronous or concerted, more concerted like fashion?
Yeah, so we think like under force, it will first like break the weakest bond, which is the bond that is between the two attaching group. And so once it breaks, it will form two radical species, and which is not stable, then drives it to isomerize to this tricycle octadiene product. So it's pretty much straightforward mechanism here.
And I would say there's some experimental evidence that strongly supports that. That involves some single molecule force spectroscopy kinetics. And the details are probably more than anyone wants to hear right now, but the upshot is that from those characterizations, we can get a fairly precise idea of where the transition state of the rate determining step in the mechanical reaction is. And that position corresponds very nicely to the scission of just that first bond rather than like a more concerted process.
I have a couple of questions about applications, especially with the fluoride releasing. Have you imagined how the spatial variation, where there might be greater force in certain regions could be used to, say, selectively etch maybe glass or some other, I'm just thinking that the reader, the participant is really trying to understand, what's some of the potential applications that might be really unique to these kinds of materials.
Yeah, so that's a great question. So I think like all of us know that HF is just so versatile reagent, and it can, as the participant mentioned, it can be useful for glass etching, semiconductor, silicon wafer, a bunch of so many useful applications. And we actually did try some applications, but the problem is that we cannot see a very efficient activation in bulk materials for some reason. So, but if we can encounter this potential limitation, then definitely we can use this HF as a reagent to remodel materials or glass etching.
And one of the things that I like about, one of the things I like about this question is that it ties down to what to me is a very fundamental intrinsic challenge, which is that I can apply a macroscopic stress across a material, but at the molecular level, there's a lot of heterogeneity in how that force is distributed. And so I think there's a real opportunity to use chemistry to enact highly local molecular level responses that deal with a molecular scale, what is fundamentally a molecular scale problem, like a lot of tension in just this bond or in this very small region of the material. And you can only do that with molecular tools, right? So this is the kind of challenge that requires chemistry and chemical reactivity to solve. And I think that's a real opportunity.
The last question, I'm going to slightly rephrase it just a little bit. I think this is basically where it's going. And it's a really interesting question about the connection between polymer mechanochemistry and small molecule mechanochemistry. And really the idea, I think, behind the question is there a way that you can imagine using the mechanical energy in a maybe polymeric material or other material to ultimately have an effect on a, say, small molecule reaction? And so I'll just, I'll leave it at that and let you try to answer that. So anybody, I think on the group here can take a crack at that.
Yeah, maybe Li Chi, you can talk about kind of part one of your dissertation work along those lines.
All right, so let me just rephrase this question. So the question is, what is the correlation between the polymer mechanochemistry and small molecule, right?
Or how could it be used for? Was it more like how could it be used for small molecule synthetic chemistry applications, like catalysis, for example?
That's right.
Yeah, sure. So like one of my PhD project that I haven't mentioned here is actually understanding like how force affects the elementary step of the catalysis, which is the oxidative addition. So at that point, like you can't really use the polymer mechanochemistry to it, but instead you can use the molecular force probe that mimics the force that are applying to those microscopic step and understanding how force affects the rate of that step. So yeah, I'm not sure if Steve has any more idea on that.
Yeah, but I think this is one, for me, one potentially very exciting opportunity, right? So that because force can be applied transiently and essentially switched on and off fairly easily, can I use it as a way to change the state of a catalyst here? It could be a reactant, right? But to essentially toggle between states that, for example, might be optimized, each of them to a different elementary step in a catalytic cycle. There's a long way to go before we can connect and get all the way there, but I think that's one example of an opportunity. I think there are other opportunities in synthesis here that that will probably be empowered not just by insights into mechanochemical reactivity, but really into some development for ways to more efficiently and evenly distribute forces across the reactants of interest.
We have to end it here. Thank you all. So before we're going to end, before we end, we're going to have a surprise question, and I want to bring back actually all of the speakers, and Steffi is going to ask the surprise question. But Thieme has been so bold in terms of thinking about, where is the field of organic chemistry moving? And I know that she's intrigued to ask all of you an important question that really gets at this broader picture of the relationship between what you're doing and sort of fundamental organic chemistry. So Steffi, I'll let you ask the question.
Yes, thanks. So first of all, also thank you for the last speakers for giving a wonderful talk. Yeah, my question to all of you is what's the most common misconception about organic chemistry? And we would love to hear your answers. You can answer as a group, or every person can answer. And maybe we find a person who wants to start.
I'm going to call on James, because I think he already kind of set it up in some of his intro talks. So I'll let him get started. How about if we let James get started?
Happy to. The biggest misconception about mechanochemistry is that it's not chemistry. It's some magical process that happens, but it's all bonds that interact. And so how they interact, why they interact, what energetics we use to make them interact is almost irrelevant. There's some relevancy in terms of some of the cultural things and the assumptions that we make over a period of time. For example, the biggest one that I point to is pH. So something that's really, really simple. By definition, it has to be in some liquid in order to make that work. So if you don't have that, then what is an acid or a base and all these different things? And we make these assumptions. And what I tell my undergraduate students in the classroom is sometimes over a period of years, you forget the assumptions you made and then they become fact until you do something different.
Okay, next one.
I think Steve's going to go next.
Yeah, I'll take crack at this, and certainly it's a little intimidating, right? I don't want to speak on something so broadly. I'm sure people have different views, but I might say a broad misconception about organic chemistry to me is that it's a body of knowledge that you're expected to have, rather than a set of principles you're expected to explore. And I think there's some historical reasons for that, from sort of a century plus ago, but I think there's still aspects of that especially get communicated early on in the organic chemistry curriculum, I think often very unintentionally, but that would be my answer.
Thanks, Steve. Jeff, I think you also wanted to answer. That's perfect.
Oh, what I saw today was really hypothesis-driven research, and it is, just asking questions that are asked in organic chemistry as well. But oftentimes, once it's very much aligned with the answers we've already heard, but you start to assume that knowledge becomes embedded in the way that it is just this way, and suddenly when you start to look at, say, Leche showed a very beautiful example of force-free unreactive difluorocyclopropanes versus that same reaction that could be mechanically triggered to undergo. And so I just think it's asking a question differently, and so it's framing a hypothesis and going about testing it. And at the end, it is bonds and atoms that really make the common ground that we all work on, and we all have the underlying potential energy surface of the molecular species, and can we manipulate it in interesting new ways with force? And Steve's group has done great. Robert, you or any of your trainees might like to comment on this as well.
Yeah, so I mean, for me, there's one common misconception that maybe that's a controversial opinion. At least sometimes when I talk to, let's say, the more old school professors, is that the polymer chemistry is different from organic chemistry, which it sometimes is in the physical chemistry department, right? But oftentimes I get asked whether I'm a polymer chemist or I'm an organic chemist, and in the end I have to say I make molecules that do something, yeah. And that's the way it is, and this kind of makes it hard sometimes to talk to people that wanna push themselves into a specific community or anything. So I guess. That is something in the end. Being a synthetic chemist is using all tools from all kind of disciplines and subdisciplines. Yeah.
And I'd love to hear from at least one or two of the trainees. You just came through these organic chemistry courses that we were talking about, and so you're now thinking broadly about the field of organic chemistry, and it would be very interesting to hear what your thoughts are.
Yeah, Jeff, if you'd like, I could chime in on that. And I think that, I mean, the most I can do is echo like the really great responses I heard from all the other panelists. But I think to answer that, I could share an anecdote that happened when I was taking undergraduate organic chemistry. I was in the library late night studying for organic whatever, and we were playing around with modeling kits. And I was holding cyclohexane in my hand, and I was trying to figure out how to make a certain reaction work. And sort of in frustration, I grabbed it and I ended up breaking it. And I said, why can't we just do that with molecules? Like, why can't we just break them? And then I remember someone across the table saying, that's not how chemistry works. And then 3 years later, I joined the mechanochemistry group. So the point I want to make out of that is similar to what has been echoed already, is that mechanochemistry, from my current limited experience, what it strongly seems to me is that it's not some form of strange voodoo chemical reactivity that a lot of that a lot of my friends who are not in the field think about it. It's really chemists who are trying to find ways to expand what they understand and believe to be chemical reactivity itself in new and exciting ways, in materials, polymers, etc. And so to me, it's just this continuous exploration of and redefinition of what we think and what we believe to be chemical reactivity.
Yeah, I totally agree with Jaffer. Mechanochemistry here is kind of seen as like a sustainability tool to get rid of solvents and use it in that way. And so whenever we present our research and we talk about isolation of the product and how we get yields, and sometimes we have to do liquid assisted grinding for some of the things that we do, and others be like, well, we thought you were green. I thought it was the whole green thing. And we like, the understanding is more so highlighted, and we can take a different tool or we can take a different method to understand, chemical reactivity of organic synthesis in mechanochemistry that is different from traditional chemistry. And so a lot of people like to get hung up on, well, I thought you were trying to, be green or use no solvent or something like that. And like, yeah, that's a point, but we try to get you to understand, like, this is a method, this is a different method that we are trying to employ to understand organic synthesis in a whole different field, in a whole different way. And I think that is my experience of the misconception of mechanochemistry, for sure.
All right, Steffi. Please, David, please go ahead.
I would also connect to what has already been said. Basically, I think I also sometimes see when I try to maybe explain mechanochemistry to people, this has been seen as something which is a bit weird or different way to activate some chemical activities. But what is maybe missing is this idea that what mechanochemistry is, and in general, what is missing is this view of this multidimensional energetic profile of molecule and structures. And this is sometimes, there's this old school of view of this energetic profile in multidimension as something which is static, but in the end is something which is very dynamic and can be modified and explored in a multitude of ways that then you can find a very different way of how molecules can interact, react, be modeled, and perform different reactivities. So this is something that this multidimensional energetic profile is something which is very dynamic and can be explored in the different, very different ways, I would say.
I think at that, I'm going to put a wrap on this. So Steffi, I think what you heard today and all the participants really is a great snapshot of where the field is at right now. In chemistry, it's really about the intersection of matter and energy. And there are just few forms of energy that we can bring: thermal, light, electrochemical. But really the one I think that has been least studied, at least in terms of trying to achieve the kind of selectivity of reaction that we want to achieve in chemistry, is that of the use of mechanical activation. And so the field is moving forward. I think it's really uncovering some fundamental principles. I really thank Thieme for being able to be so bold as to look at how this strong connection exists between mechanochemistry, organic chemistry, and how we begin to think about reactivity in a more general sense. So thank you so much for being the organizers of this webinar.
Great. So yeah, thanks also to all speakers, also to the participants for taking the time for this long event. I would also like to thank my colleagues in the back who really did a great job. Yeah, and we are looking forward to the next Cheminar series. Yeah, and I wish you now a nice, whatever, afternoon, lunchtime or whatever. Yeah, and see you soon. So goodbye and thank you.